Hello, and welcome to this webinar titled Transformational Science, Moving from Challenges of High Concentration Protein Formulations Development to Meet the Needs of High Potent Bispecifics, hosted by Biopharma Asia Magazine and presented by Sachin Dubey, Ph.D., Head of Formulation and Analytical Development at Glenmark Pharmaceuticals SA. My name is Stephen Edwards, and I'll be your moderator. Now please allow me to introduce our presenter. Sachin Dubey is presently working with Glenmark Pharmaceuticals, Switzerland, where he is heading Formulation, Analytical and Drug Product Development Unit. His current responsibilities include designing and executing product development and characterization strategies for both early and late-stage products for Glenmark Biologics. Sachin earned his Ph.D. from the University of Geneva, Switzerland, and has previously worked with Novozymes Biopharma, Denmark. He has over 12 years of experience in biopharmaceutical formulation and analysis. His principal research interests are protein formulation, stabilization, combinational product development, analytical characterization, DOE, and QBD. He also has a keen interest in practicing lean principles. He has received multiple research awards and has several publications and presentations to his credit. I will now be handing over to our presenter. Welcome, Dr. Dubey.
Good afternoon, good morning, and thanks, Stephen, for your kind words. Today we are here to discuss on the challenges we are facing when we are developing highly potent bispecifics. And before really we go into highly potent bispecific, or in other words, low concentration biologics, the industry have already observing high concentration protein formulation and challenges associated with it. There are multiple challenges which are related to aggregation, viscosity, particles, and so on and so forth. But before really we go in terms of what is high and what is low, I just want to make sure that these probably can be considered today as a related term. If we consider high concentration, for example, 20 years back, anything which is around 50 mg in the field of biologics might have been considered high concentration. 10 years back, somewhere around 100. Currently, we might be in the range of 150 to 200 milligrams per ml of protein to be considered as high concentration. The same is true for the low concentration as well. It's a subjective definition, but for the sake of presentation today, we can consider in general sub-milligram levels to be low concentration, and we are going to have a look on what kind of different challenges they offer and the opportunity they offer to overcome from a formulation and analytical perspective, or in general product development perspective. So now starting with low concentration biologics, they are not new. Some of the listed on the slide are interferons, growth factors, and a series of insulins which are there for more than 20 years. And they have been successfully developed. All of them are in sub-milligram levels. However, most of them, if not all, are injected subcutaneously. The challenges is aggravated when we had to develop an IV infusion system which essentially further dilutes the product from, let's say, milligram or sub-milligram levels to nanogram levels. And this is essentially the case or the trigger from by virtue of bispecific which are in development. The slide here reference from Drug Discovery Today 3-4 years back summarize very nicely different formats which are in development in terms of bispecific and potentially trispecifics in different clinical trials. From a listing point of view, the next slide captures various different bispecific which are in clinical trials along with the different stages of development. And if you see the first bispecific bispecific which got approved was from Amgen, blinatumomab, Blincyto, CD3 redirector, approved in 2016. And a series of them are there in development. A total of around 130 bispecific are ongoing in different stages of development from phase 1 to phase 3, either in a different format or in various different indications. The reference currently from this one is a very recent December 2018. There may be a couple of more which are new in the development, entering phase 1 potentially. Now, Glenmark also have a specific proprietary BEAT platform, which stands for bispecific engagement of antibody based on T-cell receptor. We have certain advantages from engineering perspective. In very simple terms, these are engineered molecules to force the heterodimerization inspired from nature, inspired from T-cell receptors. There are a series of advantages which are beyond the discussion for this presentation sake. However, the key point here is that these are extremely potent. As for any other bispecifics, typically bispecific are around 1,000-fold or 10,000-fold more potent than standard molecules. And by virtue of this potency, the dose requirement is in the similar ratio, around 1,000 to 10,000-fold lower than standard mAbs. And this is exactly the reason of various different challenges. For the sake of this presentation, I'll categorize them in 4 major challenges into analytical, formulation, clinical, and manufacturing. Some of them we will find overlapping. So starting with analytical challenges, this slide here is a non-exhaustive list of various different analytical tests which are very typical for a standard monoclonal antibody. And all the tests in the last column listed or highlighted with yellow are those which are sensitive to concentration or are influenced by concentration. They typically fall into, of course, the concentration itself, so the product content will certainly be influenced by concentration, but purity, like size exclusion chromatography and capillary gel electrophoresis, charge profiling, peptide mapping, and to some extent also in some cases efficacy, and I'm going to discuss some of those with examples. Starting with size exclusion, chromatography can be considered as a workhorse. I think most of the regulatory packages for any stage of development include size exclusion chromatography, specifically when it comes to monoclonal antibody or bispecifics. They are, as I mentioned, they are always present. They are among the key purity assays. The main function of these assays are to differentiate between monomer and dimer plus trimer and aggregates and so on. So in order to quantify what is the percent of monomer is the main purpose of this assay. And if the concentration of your product is on a lower side, the ratio of your product to the excipient will be, let's say in the word, non-favorable from analytical perspective. As it is depicted in the chromatogram here, you see an increase or you see an interfering peak around 5 minutes, and when you run blank buffer, you will observe the presence in the blank buffer the same peak or similar peak as well. This is also represented, or this is also pointed out by others as well that polysorbate 80 can influence insect based on the ratio available. Now there can be various different solutions available to it. Diluting it further, making the detector more sensitive and selective, like for using fluorescent detector instead of a UV, can be solution, or the use of twin trap or polysorbate trapping column can be the solution. In this case, we have used and successfully demonstrated that if we use twin trap column, this interfering peak, which were earlier present around 5 minutes, are not only removed from the product but also from the buffer itself. Moving on to the next assay, potency. Again, potency can be measured in different format: ELISAs, competitive ELISAs, or cell-based. All of them are having specific characteristic of this S-curve, which have 3 or 4 main characteristics: upper asymptote, lower asymptote, and then the linear region. And if you have highest concentration, which is not very far from your starting concentration of the assay, you can see on the extreme right an interference from the excipient. This is typically known as Hooke effect and can influence the upper asymptote and influence the ultimate result from quality perspective. There are 2 approaches typically which can be used, or let's say two easy approaches which can be used to fix this. First approach could be to use your reference standard in the same buffer, or essentially dilute your reference standard first in the same buffer as your product is in. Or the second approach, which we have chosen here, is to use a buffer exchange column. And if you see the right hand graph, it clearly depicts that if you buffer exchange your sample, the effect of interfering polysorbate, again in this case, is negated. Moving on to the next challenge, of course, protein content or the concentration of the API is a key parameter. In protein pharmaceutical, most of the time absorbance at 280 nanometer is considered to be the key assay to analyze, quantify the content of the product. And depending on the ratio of the excipient to the product, signal can be influenced from the buffer itself. I will just present you a couple of examples. These are 2 different buffer systems, buffer 1 and buffer 2. And if you observe on the left hand side, buffer 1, already at T0, there is a background absorbance just coming from the buffer around 0.15 absorbance unit for A280. Plus, in addition, if you see different stresses for this buffer, there is a lot of inconsistency for this buffer. On the other hand, if you see buffer 2, this is much well behaved, but still there are some red flags on how to use this buffer. And this becomes extremely critical when we are developing low concentration formulation, because any change in buffer background absorbance can influence ultimately the concentration of your protein, or under measurement of the concentration of your protein in the final formulation. There are different approaches to overcome this challenge. Using appropriate blank is certainly required if you are going with A280 measurement. It is also recommended to put the blank buffer on stability as well, in order to have similar batch all across the stability. Use of an increased path length can be an additional solution, which increase the sensitivity of the assay. We can use, instead of 1 ml path length, 2 or 5 ml, or even longer path length. Alternative techniques like HPLC, solo VPE can also be made available. They may not be as straightforward as using a spectrophotometer, but depending on the complexity, these can be used as well. Yet another analytical challenge is the presence of homodimers. So for bispecific, it is important that there is a presence of heterodimer. That means this format, which is in the middle of the picture on the right hand side of the slide. But by virtue of association, we can have homodimer of A, or you have homodimer of chain B. And obviously, in your bispecific product, you want to limit those. They can act as variant or impurity depending on the biological and toxicological profile of these homodimers. And again, depending on the format, we may require very sensitive, extremely specific techniques like MS, even for a routine basis, for characterizing and understanding different differences between homodimer and heterodimers. Again, still on the analytical challenges, there is a list of assays, CGs, ICE, CEX, peptide mapping, which may require additional sensitivity, and in general, for all of them, the solution or potential solutions could be use of either sensitive detector instead of UV. Most of these on a standard basis come as a UV, but now there are techniques coming with fluorescent detector. For example, CG system in Morris, you have fluorescent detector possible, or otherwise it's cumbersome, but you will have to pool multiple product vials in order to generate reliable data for these techniques, these properties. Moving on to the next challenge, formulation. The challenge is coming from formulation development. In this particular case, from formulation perspective, the very first challenge is to balance the desire. On one hand, from clinical dosing, the clinical people will look for the as low as possible nanogram per kilogram, which converts into nanogram per milliliter concentration for clinical dosing. On the other side, the challenge is from analytical limits. You cannot have a formulation in those nanogram per ml if we are not able to release them, if we are not able to analytically characterize them. So the limits could be in the range of microgram per ml. And from formulation perspective, it's a balance of analytical limits and clinical dosing. Yet another challenge is the chemical reaction, or essentially the purpose of formulation challenge is to ensure that your product is stable for the intended period of stability, intended period of use for the product. And so essentially you have to control and understand and limit the rate of degradation. And if we see these two green boxes are of same size, and if we see on the left-hand side box, we observe that whether we consider products with the red line on the left-hand box, you see a very clear, very evident drop in the red line. On the other hand, if we see on the right-hand box, the difference is much less. So in other words, the impact of these small changes is very difficult to pick up, and could be very difficult to understand, especially considering the analytical variability of the available assays, especially during early stage of development. Yet another challenge is that typically from formulation perspective, you would like to have one composition for your bulk drug substance and same composition for drug product. As I mentioned, for drug product, you would like to go as low as possible, whatever your analytical allows. But BDS, you don't want to dilute too much. You want to have still an operationally feasible concentration, let's say, for example, 10 milligrams per ml, so that you don't have huge volume to handle and store and so on and so forth. So you need to develop a composition on a high level which covers up these ratio, these stabilizing factor for both BDS as well as drug product concentration. And I depict here in this slide a hypothetical case where a BDS and a drug product for different excipients have been presented. And if we see for BDS, the ratio and compared to DP for anything, for example, surfactant, this has a 300-fold to 1,000-fold difference. So you need to develop a case where we have same formulation, sufficient excipient present for your drug product as well as BDS. The other way around is still possible, but the challenge is that then your protein will be exposed to unnecessary additional stresses of, for example, change in osmolality factor, conductivity factor, pH, etc. Moving on to the next challenge, challenges coming from manufacturability aspects. For biologics, filtration is more or less can be termed as an obligatory unit operation. This is the operation whereby we achieve sterility for the biologics product. Filtration as a unit operation is influenced by various different properties, some of them coming from filter itself and some of them coming from the solution we are filtering. One of specific is the concentration of protein, and this becomes absolutely important when the product is at very low concentration. This graph depicts in a nice way what happens if we do not consider to if we just start filtering and filling the vial straight away. If we start filling straight away, what we observe is that in the initial few fractions, you will lose some protein. The protein is lost, the protein might be absorbing onto the filter and is not filtered out, meaning that either we will have to develop the filtration, optimize another filter, use another filter membrane, or a simple solution could be to discard initial fractions and then to homogenize further and before filling can be a solution. But again, to emphasize, for low concentration product, this is an absolutely important study to be performed. Yet another challenge from manufacturability perspective is more from theoretical point of view at the moment, whether to use a RAPS system or an isolator. The challenge here, they of course both of the systems have their advantages and disadvantages, but particularly talking about low concentration, the key aspect is, and specifically for lyophilized product, the key aspect is what happens if a vial is broken, and how will the manufacturing site will handle from health and safety perspective. How will we make sure that there is no cross-contamination? How will we make sure that there is a strong enough cleaning in place available for any of the system, whether it's RAPS or an isolator, well established for your product? These kind of challenges are extremely important to be discussed with the manufacturing guys before we start developing and consider the manufacturing aspect of these low concentration potent biologics. Potentially we can learn from some of the lyophilized oncology products from low molecular weight small molecules. The last category of challenges is coming from clinical or dosing perspective. Whenever you have a potential project, the team start thinking about first in human. Everyone is excited about it, but from formulation perspective, what we really have to think about, how are we going to dose, how are we going to make sure that this product reaches to the patient as it should be, as it is intended to be. Again, taking a hypothetical case here, let's consider in this case you have in the vial lyophilized product or a liquid product which is at 100 microgram per ml concentration, analytically feasible concentration, but your first in human is in the range of, let's say, few nanogram, 5 nanogram or 10 nanogram per kilogram of patient weight. For a 50 kilo patient, you will require anywhere around few hundred of nanograms, and this require dilution to as low as possible in order to ensure you infuse sufficient volume into the patient. This can be done either by using a standard infusion set, saline bags or a dextrose bag, or by using appropriately diluted syringe infusion pumps. In any of the cases, all through during its journey after dilution, the product is exposed to various different surfaces: glass, rubber, polyethylene, PVC, any other polymer, stainless steel in the infusion needle, and so on and so forth. And at these low concentration, different surfaces are really not the friend of these low concentration product, because at these low concentration there is always a risk of adsorption, and not only that, because of this adsorption you may denature your product as well, render them ineffective. So what you have to ensure at first place is of course we have an analytical technique whereby we can quantify in these low nanogram level, and if we see, of course, at these low nanogram, 10 nanogram per ml, there are no other analytical tool which we can use except for ELISA. But even if we see ELISA, especially in this particular case, for standard mAb, you will see that low nanogram is more or less towards the to the lower part of your S curve, and you have to be thinking about developing or putting additional points in this range, or to convert this S curve into a more linear curve by increasing the additional sampling, additional standard points in this low concentration range. So essentially going from microgram per ml concentration range to nanogram per ml range. This in addition provide an advantage of not only quantifying but also making sure that your product is retaining its activity. Ultimately, you have to also ensure that as soon as you dilute this product into saline bags or dextrose bag, which are commercially available, you do not lose those. And if you see the first column on the left hand side, if you have let's say improperly selected dilution system, saline or dextrose or anything else, you have a risk of losing product anywhere between 50 to 70%, and you may require to develop this further based on composition, based on physicochemical properties, add additional excipients to ensure that you recover your molecule and ensure the delivery of your target 10 nanogram per ml. With this, I would like to conclude with following messages. Bispecifics are becoming more and more important. As I mentioned, around 130 of them are in various different clinical stage of development. Low concentration and specifically for IV administration offer challenges of different nature. We have discussed most of them, if not all of them. And These challenges are there which can be overcome by using various different strategies. It's very important to plan it well in advance, and it's important to understand from manufacturability perspective, from first in human perspective, and design those experiments very carefully. With this, I would like to thank my team, and will be happy to take questions here.
Thank you. As Dr. Dubey just mentioned, we will begin the question and answer segment of the webinar. But once again, I would like to remind our audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, what are the different analytical tests employed for in-use testing?
Thanks for the question. So different analytical tests typically should be, but this is the final product which is essentially entering into the patient. And from a quality perspective, you would like to ensure that your product is of as high quality as you would like to. So I will recommend that you should select the test as close to as your release testing panel. Of course, you can do risk assessment for some of those, like charge profile, like fragmentation, for example, which we believe cannot be influenced during this small period of infusion, which can last from, let's say, few hours to 1 day or so. So I will propose that we should use safety-related tests. Example, microbiological burden. This is an important test. So visible particle is important, and the most important and the most ultimate is the concentration of the protein, which you want to make sure is present what you are infusing into the patient.
Thank you for that answer. It seems, unfortunately, we don't have any more time for any questions, but if you do have questions, feel free to send them over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers to you. Before we finish the webinar, I would like to ask our presenter, Dr. Dubey, if you have any closing remarks.
Yes, absolutely. I think this is an exciting area which is going to see a lot of development. There is only one bispecific which is currently approved. There is a lot of let's say unknowns which are there. Lot of upcoming challenges which are there, and those require careful planning, and in fact also for some of those situations potentially regulatory consideration is also important to ensure that we are getting onto the right track and developing the product in line with the regulatory expectations.
Well, thank you for that, and I would like to take this opportunity to thank our presenter once more. That was Sachin Dubey, Ph.D., Head of Formulation and Analytical Development at Glenmark Pharmaceuticals SA, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, as a reminder, please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.
Thanks.